Okay, I want to start by giving all of my conflicts. I have actually a lot of different companies that I work for, have started, own stock in, listed here: Petra, Agios, EPI Pharmaceuticals, Cell Signaling Technologies. I've not been on any speakers bureau, so I don't talk for companies. I have grant support from Petra, one of the companies I started, but mostly from the NCI, Gray Foundation, Breast Cancer Research Foundation, Stand Up to Cancer, and I'll talk some about the Stand Up to Cancer work that we're doing, and AACR. And I'm stockholder in the companies that I founded. I have done an honoraria for Sanofi and Novo Nordisk in the past year, but those are the only two companies, and I won't talking about any of the drugs made by any of the companies on this slide. So I only consult on things that I don't know anything about. So I have to say that the last course I had in biology was in 1964. So I'm entirely a chemist. My undergraduate degree was in chemistry, my PhD in physical chemistry and biophysical chemistry, and I avoided taking any biology courses in college or in graduate school. So as a consequence, I had no prejudice about how biology works. I just assumed that it didn't disobey any law of chemistry or physics. And that actually has served me pretty well. But I did get interested when I should say I worked as a PhD student. I worked on how you convert a chemical gradient, an electrochemical gradient, into a high energy molecule. In other words, how do you make ATP from a proton gradient in the mitochondria and chloroplast? So that actually is when my first movement into something somewhat biologically relevant. But as I started my laboratory at Harvard as an assistant professor in 1978, I became interested in how you get molecules in and out of cells, just some very simple questions. How do you get glucose in a cell? How do you take up sodium or push out sodium, move in potassium? And in the course of trying to understand that, I got even more interested in how hormones and growth factors, hormones like insulin, alter the ability to take glucose up and amino acids up into cells. And at that time, insulin signaling was a total black box. We knew what insulin did. It would drive growth of cells. It would drive glucose uptake, et cetera. But we had no idea how that worked. So I began to focus on that question. How does insulin stimulate glucose uptake? And since it's an event that happened at the plasma membrane, I thought, well, maybe some lipid is involved. And I got, for reasons I don't have time to get into, I got interested in the lipid phosphatidylinositol. So phosphatidylinositol gets phosphorylated at the 4 position and the 5 position of the inositol ring. Those are those 2 circles in black with the P. Those are phosphates. And that was discovered actually the year I was born, 1949, that when P32 was added to tissue, P32 inorganic phosphate was added to tissues, some of the radioactivity ended up in the lipid. They looked to see what it was, and it was this lipid, this doubly phosphorylated lipid. And so this, in fact, was thought to the only 2 forms of phosphorylated phosphatidylinositol known to exist in 1988, before we discovered this enzyme, were PI4P and PI45P2. In other words, phosphorylation at those 2 sites that are black Ps. What we discovered was that there was an enzyme that co-purified with the insulin receptor, but it also co-purified with a variety of other growth factor receptors. And importantly, it co-purified with virtually every oncoprotein that we purified. So all the viral-induced oncogenesis, viruses that caused cancer, the Rous sarcoma virus, polyomavirus, that caused cancers in chickens and mice, when we isolated the oncoproteins, the enzymes or proteins that were made by those viruses to transform the cell, and looked at them, we found this activity was co-purifying with all those proteins. But it was also co-purifying with the insulin receptor. So the bottom line is, the insulin receptor makes a lipid, this triply phosphorylated lipid shown at the bottom, which I call an oncolipid, if you will, a cancer-causing lipid, as its normal mechanism of action. We now know that everything that insulin does in every tissue in your body, well, probably everything, we've yet to find an exception, goes through making this lipid. But all these oncogenes that transform cells also manage to turn up the level of this lipid. So even by the late 1980s and early 90s, I was pretty convinced there was some dramatic connection between insulin and cancer. So what might it be? So the bottom line is, and I won't go into all the details of the signaling pathways that we've worked on for the last 30 years to explain this, but the bottom line is what we now know is that this lipid has to be made in order for a cell to grow. And if everything is working normally, then when a child eats, or a young fly eats, or a young worm eats, or fish, or anything, this is conserved in all multicellular animals, then that stimulates the release of insulin. Insulin goes to various tissues, activates PI3K, this enzyme, produces this lipid, and this lipid then signals a number of events that tell the cell to take up glucose and to drive anabolic processes, and that's how animals live. If you knock this enzyme out in flies or worms, or in the case of animals, we've knocked it out in the heart of mice, in other tissues, muscle, skeletal muscle, fat, etc., liver, the consequence is insulin doesn't work in that tissue anymore. The cells in that tissue are smaller, and they become acutely insulin resistant. Now, as long as everything is working normally, this lipid is only made when insulin is high, and it tells the tissues what to do, whether it's to grow in a growing organism or in a mature organism. Insulin still works, but in this case, not to tell the cell to grow, but rather to tell which cells should take up the glucose, and to tell muscle and fat to take up the glucose, and to tell liver to quit making glucose and store it as glycogen. So there's somewhat different effect of insulin during development and insulin in adults. Now, there has to be a way to get rid of this lipid, and we didn't know until 10 years after we discovered PI3 kinase how the cell gets rid of it. And it was discovered in about 7 years after we discovered PI3 kinase. A gene was identified called P10 for phosphatase on human chromosome 10 that we now know is second only to P53, the notorious tumor suppressor gene, as the most frequent event that happens in all of human cancer. So this gene was positionally cloned as a tumor suppressor gene. But eventually nobody knew what it did. It looked like a phosphatase, but it wasn't clear what its substrate was. And Jack Dixon's laboratory, Jack being a wonderful biochemist, sought a substrate and tried PIP3, and it turned out to be dramatically the best substrate for this enzyme. So P10, the most frequently, after P53, the second most frequently mutated tumor suppressor gene, works by getting rid of this lipid. So as long as P10 is functional and PI3 kinase is only turned on when insulin is high, then this PIP3 is only generated briefly in various tissues and then goes away because P10 gets rid of it. But if you lose the ability to turn it over with P10, or there are activating mutations in PI3 kinase, or the gene for PI3 kinase is amplified, so there are many, many copies of it, then the level of this lipid goes up, and that drives cancer. So that's what cancer is. The majority of cancers work by this mechanism. So there turns out there are actually 4 different PI3 kinase genes that encode enzymes that make this lipid. So the B cells and T cells do it differently from the liver and muscle. They use a different gene called PIK3CD, and that gene also drives not only the growth of normal B cells and T cells, but it also drives the growth of lymphomas, the result from B cells and T cells. So drugs targeting that enzyme have already been approved. They work quite effectively in blocking the growth of those tumors. But they're different in that those enzymes are not regulated by insulin. So the only genes where you actually see mutations in PI3 kinase, the only gene, is in the one that responds to insulin. PIK3CA is the name of the gene. So the other three still drive growth, but they don't respond to insulin, and they're virtually never mutated. So that's telling us it's really insulin. And what those mutations do is increase the ability of that enzyme to respond to insulin. So those genes, those mutations are not really by themselves activating. The enzyme activity is not much different in the mutated enzyme versus the wild type, except now it requires less insulin to activate the mutated genes. So we can then ask, now, the TCGA, thanks to TCGA, more than 20,000 genes, 20,000 human tumors have been sequenced. Every gene in 20,000 patients with tumors have been sequenced. So we have a vast amount of data now that we can go in and interrogate. So if we go in and ask, well, how often do you see these activating mutations in PIK3CA, which are indicated in green on these bars, or loss of function mutations or deletions of P10, which is the light blue and dark blue, or amplification of PI3 kinase, which is the red. And then the sum of all of those events is shown at the top of the bar. And what you'll notice right off is GBM, that's glioblastoma. More than 90% of glioblastomas have either a loss of function of P10, shown in blue, or an activating mutation in PIK3CA, the insulin-responsive PI3 kinase. Uterine cancers, more than 80% have, and most of those are activating mutations in PIK3CA. Lung squamous, renal, bladder, breast, all the way down the line. So all the cancers that cause the majorities of deaths in the United States are driven primarily by mutations in these two genes, just two genes. Everybody's heard probably of KRAS, an oncogene. It's still, it's what dominates in lung cancer and pancreatic cancer. It's not as frequent as PIK3CA in mutations, if you count all types of cancers together. And I should say, by the way, the mutations in KRAS allow it to activate PI3 kinase better. So that's also driving through PI3 kinase. Now, it turns out that the same mutations that we see in cancer that enhance the sensitivity to response to insulin can occur as a mosaicism during development. So during embryonic development, if a single cell in the embryo picks up that same mutation we see in cancer, then all the progeny of that cell, all the cells downstream, have the ability to respond to insulin better than other cells in the embryo. And as a consequence, all the cells that drive that lineage outgrow the other cells in the body. So if this happens very late, just in the tip of the toe, then you just get an enlarged tip of the toe. If it happens earlier, you might see an entire leg or half of the body with this massive growth enhancement. If it happens in a neuronal cell, then you get that's in the brain, then you get a cluster of enlarged neuronal cells that respond to insulin far better. Does anybody have a guess as to what disease that might cause? Epileptic seizure. So you now have insulin being able to drive the response of those neuronal cells far better than any other tissue. And that, for many years, we had no idea what caused these epileptic seizures, but often they were surgically removed. So you can go in with a needle and remove that part of the brain, and the seizures would go away. But about 7 years ago, people started sequencing the genes in that region and found that they were different from the rest of the genes in the body. And they turned out to have the very same mutations as you see in this leg. And these various diseases, and were all called different diseases because they occurred in different tissues, they were given different names, but they're now all called one disease called PIK3CA-related overgrowth syndrome, or PROS. And PI3 kinase inhibitors are now going into clinical trials to treat these diseases, because otherwise surgical intervention is the only way to do it. So how do we try to block epileptic seizures? We already heard about this, right? Ketogenic diet. Why does it work? Lowers insulin, right? And since these mutations enhance response to insulin, it makes perfect sense why that works. but we figured it out empirically without knowing why it works. I think now we know why it works. Okay, so, 100 years ago, or 90 years ago, 95 years ago, I guess it now was, a German scientist named Otto Warburg made the observation that if you mutagenically induced a cancer in a rat, and then looked at glucose uptake into that tumor and glucose metabolism in that tumor compared to the tissue from which the tumor emerged, that the tumor was taking up glucose at a much higher rate, and it was driving anabolic metabolism at a higher rate. And this was called the Warburg effect at that time. Warburg got the Nobel Prize for figuring out how mitochondrial metabolism works, not for this observation, but he's most remembered for the so-called Warburg effect, that cancers were different in how they metabolize glucose. And in fact, back about 30 years ago, with the insight that this was pretty common, most cancers have this Warburg effect, fluorodeoxyglucose, a radioactive form of glucose, was developed as a way to try to determine where the cancer is in the body. So FDG-PETs, I think probably everybody in the room has heard or have a relative or someone who's had an FDG-PET. That's why it works, because you've activated PI3 kinase pathway, and that stimulates glucose uptake into the tumor. So this is an example of a woman who went on a Stand Up to Cancer trial. I should say that Stand Up to Cancer AACR provided me and my team a $16 million grant to try to work with industry to figure out how better to develop PI3 kinase inhibitors as they were going into phase 1, phase 2 clinical trials about 10 years ago. And so I was really, again, not being a medical doctor, not knowing anything about biology. Obviously, I had to have a really good team. First thing I learned in science is you must make sure that everybody you hire is smarter than you are. It makes your job easier. You don't have to tell them what to do. So that's what I concentrated on. I got some of the best clinical trialists in the country working on our team, and we worked particularly with Novartis to try to figure out how to, since they had some of the first drugs going into clinical trials, how to best make their drugs work, either by changing how they're delivered or doing drug combinations. So this is one of the earliest patients to go on a trial, and the combination we wanted to do was an aromatase inhibitor plus a PI3 kinase inhibitor. Because what we'd noticed was, even then, long before the 20,000 tumors were sequenced, was that breast cancers very frequently had PIK3CA mutations, and the subset that had them were the ER-positive breast cancer patients. So we had drugs, aromatase inhibitors, for treating ER-positive. They were pretty effective, but a lot of patients failed to respond or ultimately became resistant to that therapy alone. So we reasoned that this might be a good trial to do. And in fact, as I say, the PIK3CA mutations were mainly in the ER-positive subgroup. And in fact, 40% of women who are ER-positive have PIK3CA mutations. And that's the most frequent subtype of breast cancer. So this was one such patient to go on to our early trial, and we combined an aromatase inhibitor, letrozole, with a PI3 kinase inhibitor, Alpelisib. And I'll tell you a little bit more about this drug in a moment. So this is a drug that's highly specific for just the PIK3CA type of PI3 kinase, the one that insulin stimulates. So you can see by FDG-PET, this woman had metastasis to the liver, multiple lobes in the liver, had tumors growing in them. She had failed aromatase inhibitor alone as a single agent. She also was HER2 positive. She went on Herceptin, failed that, did a combination of those two, still failed, and she was progressing and run out of choices at this point when she came into our trial. So we gave her the PI3 kinase inhibitor in combination with letrozole, and 2 weeks later, by FDG-PET, there was no glucose up going into those nodes in the liver anymore. Now, it turns out if we image the tumor in other ways, MRI, CT scans, then there's still tumor there. It had not shrunk. It just couldn't take up glucose anymore because we turned off PI3 kinase, the mediator of glucose uptake into the tumor. So that all made sense. That's exactly what we hoped to see. This woman ultimately, after 3 months, the tumor shrunk, and she was pretty much disease-free for about 1.5 years, but ultimately progressed. So this wasn't a cure, but it was definitely a long-term response. So we were excited about that. and went on to enroll a number of patients. Now, unfortunately, she was in the minority. So most of the patients that we put on this trial had PIK3CA mutations, and they were ER positive. And yet, some of them responded dramatically like her, but the majority didn't. And so we did FDG-PETs at 2-week intervals, before and after drug, on about 20 of these patients. And we found that most of them had either very little change at all in the FDG-PET, and some actually the FDG-PET was bigger, more glucose going on after giving the drug than before, which was kind of shocking. I'll explain to you in a moment why that was so. But that was kind of shocking to the oncologist. Not quite as shocking to me, because you'll see my reasoning in a moment. So it turned out that the patients whose FDG-PETs were like this one, of going away immediately, they all had clinical responses. Their tumors shrunk. But those who had very little change, or the glucose actually went up, went off the trial very quickly. They progressed. So this was actually an early indicator, and we actually proposed to Novartis that every patient that went on their trials, and in fact their label would be that unless you saw a change in FDG-PET in 2 weeks, you don't continue on the drug. They didn't like that idea. First of all, it costs $8,000 per PET, and I had to pay for all these PETs myself because the company wouldn't do it. But we learned a lot from that. In the meantime, we were making mouse models. Here was an ER-positive breast cancer mouse model with a PIK3CA-driven tumor. And again, you can see they're very PET positive. When you turn on the mutant gene, indicating it's really this mutated form of the gene that allows it to respond to insulin and turn up glucose uptake into the tumor. So those bright colors there are glucose going into the tumor. Here's another. This actually is a BRCA-driven tumor. Again, PI3 kinase inhibitor, you can see on the left is before giving the inhibitor, on the right is after. We don't completely turn off glucose uptake into this tumor, but we definitely slow it down, and we can see responses in this disease setting as well. This tumor doesn't actually have a PIK3CA mutation, but it still is dependent on PI3 kinase to take the glucose up. I should add that in our phase 1b trials of combining letrozole with PI3K, we did admit about 20 patients who did not have PIK3CA mutations. And about 5 of those responded, had clinical responses. In fact, very long clinical responses, even though they didn't have a mutation. So this wasn't like EGF receptor mutations in lung cancer, where everybody who has a mutation responds to EGF receptor inhibitor. And people who don't have a mutation have no response. Same thing with BRAF mutant melanoma. Only the patients who have the mutations respond. This is not what we saw in this disease. There was a skewing towards patients with PIK3CA mutations responding, but there were patients without mutations who responded, and there were many patients, in fact, the majority with mutations who didn't respond. So what's going on? Why isn't it simple like EGF receptor or BRAF? So again, getting back to the mutations, you can see that if we look at just PIK3CA, so in this graph I've left out the P10 mutations, you see it's women's cancers that dominate the PIK3CA mutant subgroup. Uterine, cervical, breast, ovarian, all have high rates of either mutations or amplifications of PI3 kinase. So when we put our standard cancer team together, we decided to focus entirely on women's cancers because that would really capture the majority of patients with mutations in the gene. But there were complications in how we would do this. As I indicated earlier, PI3 kinase inhibitors that target PIK3C delta, the one that's in B cells that drives B cell lymphomas, were very effective and got approved quite quickly. 3 different such drugs have already been approved. But they don't have the problem of being insulin driven. So they're driven by B cell receptor. And so we don't have to worry about insulin elevations affecting the response to the drug. But the drugs that target PIK3CA, the insulin responsive PI3 kinase, have been very difficult to get approved. As I said, that trial that I showed you, that was started almost 10 years ago. So what's the problem? Well, the problem is that if you give a PI3 kinase inhibitor, what do you think is going to happen to blood glucose if you inhibit PIK3CA, the insulin-driven PI3 kinase. What's going to happen to blood glucose if you inhibit that enzyme? It's going to go up, right? You're going to get acute insulin resistance, and we'll go into that in a little more detail. Now, if the insulin levels go up, you can now act, if it gets up high enough, you can override the effect of the drug, because you never can get a concentration of the drug that gives 100% inhibition of the target. There's always an efficacy toxicity problem with doing that. So if you can get insulin high enough, you can activate the remaining amount of enzyme and bring everything back to homeostasis again. Keep in mind, the PIK3CA mutations we see in cancer allow the tumor to respond better to insulin than your liver or muscle. If you happen to be somewhat insulin resistant, then the tumor has a tremendous advantage of any other tissue in your body of responding to insulin. That's why when you do an FDG-PET, the glucose is going into the tumor much better than it's going into any other tissue in the body, except the brain, I should say. But even with the brain, which there's a tremendous amount of glucose going into the brain with a PET signal, if you have a glioblastoma and PIK3CA mutations, that's still even brighter than the normal brain. So anytime you turn on, by mutation, this insulin-responsive PI3 kinase, that tissue has an advantage over the rest, every other tissue in the body. So this is now shown as a cartoon for those who don't like words. So we've seen versions of this over and over again. This is the muscle in this case, very simplified. You eat your pasta, for those of you who still eat pasta. I'm sure nobody here ever puts sugar in their coffee, so we don't worry about that. I replaced the sugar in the coffee with the pasta. You would never drink apple juice, so I assume nobody does that anymore. In any event, the glucose gets into the bloodstream either slowly or rapidly. If it gets in rapidly, it's a bigger problem. The pancreas starts releasing insulin. It activates PI3 kinase in muscle. That drives that excess glucose into the muscle. It gets stored as glycogen, and everything's fine. If you happen to be walking around, it'll actually burn that glucose to make ATP. Same thing, it can go into the brain. The brain probably at rest is using up about a third of your glucose is getting burned in the brain. So those of you who are actually thinking right now and maybe moving around a little bit, you're in good shape. So if everything's working fine, that's the way it works, and the glucose level comes back to homeostasis very quickly. We saw lots of that. If, however, you're insulin resistant or you take a PI3 kinase inhibitor, then very quickly the blood glucose goes up, insulin goes up to try to bring that glucose back to homeostasis, and as a consequence, you try to, that elevated insulin is attempting to bring everything back to homeostasis. If you have a PI3 kinase inhibitor, it's going to take a lot more insulin to come back to homeostasis than if you didn't. Or if you're insulin resistant, it's going to take a lot more insulin. So the insulin levels are going to be higher in people with insulin resistance, and then you give a PI3 kinase inhibitor on top of that, and insulin levels are going to be sky high, higher than ever seen in a human being before. And that's exactly what we see in humans who take this drug, and in mice that we give the drug to. So if, however, the tumor has a mutation in PI3 kinase, then it will respond to very low levels of insulin, and even in the presence of the drug, will still respond pretty well. That's why we're having trouble getting the drugs approved. That enhanced response to insulin means that the ambient insulin in your bloodstream can override the drug effect in the tumor, and that has been the problem. So I'll tell you about 2 approval trials that just got announced over the last 6 months. The first one was from Genentech Roche, a drug called GDC0032, which in our hands, in our preclinical models, and even in our phase 1 models with that drug, it was actually looked like the most promising drug for inhibiting PI3 kinase in the clinical setting. So it went into phase 3 clinical approval trial along with a drug, fulvestrant, that targets the estrogen receptor for ER-positive breast cancer patients, and they selected PIK3CA mutant breast cancer patients. So this trial seemed very well designed. Over 570 patients went on the trial, and the result was announced in June 2018. Now I should say, and I'll show in the next slide a little more detail about the competing trial, was from Novartis. Virtually identical trial, same number of patients, same disease setting, ER positive, PIK3CA mutant breast cancer. Same secondary fulvestrant in combination in both trials. So basically the same trial. And Novartis started their trial about 1.5 to 2 years earlier than Genentech, but Genentech finished their trial faster. So a little surprising they were able to leap ahead. The problem was the trial failed. So this was revealed at ASCO in June, and one day after the trial was presented, Roche abandoned the drug. So this is now a dead drug. Lots and lots of money spent on this over about 8 years, and it was abandoned, Alpelisib, even though it looked so promising in preclinical and early stage trials. If you looked at the data, there were some regions in the world, this was a worldwide trial, there were some regions of the world where there was zero benefit. So the overall benefit was like 2.5 months. That's not enough to get a breast cancer drug approved. And in many areas of the world, there was zero benefit, not a single month progression-free survival. So the second drug that went in trial was the one that I showed you in that patient, the woman whose PET scan went away, BYL719, now called Alpelisib. Again, same drug, same combination, again, about 550 patients or so. And the endpoint was 11-month extension instead of 2.5. So what was the difference? And I would argue, this hasn't been proven yet, but when we worked with Novartis on the phase 1b trial with this drug, I insisted that any patient that had to be managed on insulin had to be excluded from the trial. So imagine you get, so first of all, anyone who had type 2 diabetes was immediately excluded. But a lot of people were insulin resistant and didn't know it, as we've heard over and over again. You know, half the people in America are insulin resistant. So if they didn't know they were insulin resistant, they could enroll in the trial. Endocrine consultant called in on every patient for the first dose, followed them, and we watched, monitoring both the glucose and the insulin level. And if the glucose went up more than 3-fold, and I should say everybody went on metformin right off to anticipate that there was going to be insulin resistance. And if the glucose level did not go up more than 2 to 2.5-fold, then the patient could stay on the trial. If they went up higher than that in the trial that we did with Novartis, they couldn't be managed with metformin, they had to go off the trial. So we, as a consequence, something like 1/3 of the patients we tried to enroll had to be taken off the trial. And that's why it took so much longer to get that trial enrolled in the phase 3, because they maintained that requirement that if you had required insulin to keep the glucose managed, you had to go off the trial. Genentech had no such requirement. It was entirely up to the endocrinologist how to keep the glucose in control. They wanted a fast enrollment. They wanted lots of patients. And so guess what? I will bet that the majority of their patients were managed with insulin. So you already have very high insulin, and now you add more insulin, and the chances that that will override the inhibitor is remarkably likely. So this is, again, what's happening. You have, in this case, I'm showing the liver instead of the muscle, but similar story. You eat sugar, pancreas makes insulin, insulin stimulates glucose up, blocks gluconeogenesis in the liver, stimulates uptake in muscle and fat. But if the tumor has insulin receptor, and most tumors do, then it will also stimulate glucose uptake into the tumor. If you happen to be insulin resistant, then your ambient insulin is high all the time. You're insulin resistant in the liver and muscle, but you're not insulin resistant in the tumor. The tumor has a mutation to make it hyperinsulin sensitive. And so it gets all the glucose that should have gone into your muscle or fat. Hence, the FDG-PET signal, easily seen it in the tumor. On a high glycemic diet, you can imagine, it just gets worse and worse if you're on a high glycemic diet. So how do we manage this? So our postulate then is that if we could keep the insulin level low, the drug should work better because it won't override and reactivate PI3 kinase in the tumor. So this data is from a mouse, but it's actually identical to what we saw in the human trials. So as I said, in all the human trials, everybody went on metformin before taking the first dose to try to anticipate the glucose overshoot. So the blue is just normal child, no metformin. The green is with metformin. The red is a ketogenic diet, but we only started the ketogenic diet the day before. So we did no glycogen depletion, no long-term ketogenic diet. We just made sure that the time that they were given the drug, they had no rapid release carbohydrate, no carbohydrate in their food, or very little, less than 10%. The purple is the sodium glucose cotransporter inhibitor, which, as many of you probably know better than I, in human trials or human studies, can be much more effective than metformin in keeping glucose low. This drug, keep in mind, blocks reabsorbance of glucose in the kidney, and so the glucose ends up in the urine. Major side effect being yeast infections, but in fact is pretty good at keeping glucose under control. The right part is the insulin levels at the end of this treatment after 2 hours. And you can see that ketogenic diet on the right is by far the champion. In this case, this was a ketogenic diet for a week run-in before we measured the insulin level acutely after giving the mice the PI3 kinase inhibitor. So metformin is really not very effective. That's what we were using in the clinic in all the patients. Was not very effective in keeping glucose down or insulin down. Sodium glucose transporter inhibitor is better. Ketogenic diet is the best. So the question is that going to be helpful? Is it really true that if we can keep the insulin level, we can have a better effect on the drugs? And the answer is absolutely. So this is a breast cancer, PIK3CA mutant breast cancer patient, like that mouse I showed you earlier with the FDG-PET signal. And you can see that a PI3 kinase inhibitor, even though it slows down that tumor some, doesn't cure that tumor. Ultimately, the tumor keeps going. And that's what we typically see in the human patients as well. But if we put that mouse for a week on a ketogenic diet, and then begin and continue on the diet throughout the therapy, then the tumors just go away. So we've now done this in 8 different types of tumors, including AML, where we're looking for a tumor that would not respond this way. Even a single tumor would have been a nice control. We've yet to find one that doesn't show this kind of response, whether or not they have a PI3 kinase mutation. This one does, but all of them we do did not. Even pancreatic tumors that never have PIK3CA mutations just completely go away if you combine a PI3 kinase inhibitor with the ketogenic diet. The ketogenic diet by itself, in some cases, has some slowing down of the tumor. In other cases, it has no effect on the tumor. So I wouldn't say that a ketogenic diet is ever going to be an effective therapy for a tumor. Maybe glioblastoma, but it's not going to cure it. But you put together a PI3 kinase inhibitor with a ketogenic diet, and invariably in every model we've looked at, we see these kinds of dramatic effects. Here's an example of a tumor. This was actually an endometrial tumor, a human endometrial tumor organoid. So we grew an organoid out of the patient's endometrial tumor from surgical material, implanted it in an immune-compromised mouse, and then multiple mice, and we tried treating either with no drug at all, that's the vehicle at the top, and the brown stain indicates autophosphorylation of the insulin receptor. This is an antibody that recognized specifically sites phosphorylated on the insulin receptor. And you can see some brown in there. It's not a whole lot of brown, but this is without the inhibitor. If you look directly below that, that's what it looks like after you give the inhibitor. You can see that activation of the insulin receptor in the tumor is dramatic when you give a PI3 kinase inhibitor. Unless, in the right bottom panel, unless you give a ketogenic diet and then the same drug at the same dose, and then all that brown stain goes away. So it's clear that you can prevent insulin from activating the tumor by giving a ketogenic diet because it just lowers insulin. So here's another tumor. This is a triple-negative breast cancer tumor that has, from a patient, I should say, transplanted into the breast of an immune-compromised mouse. It has a PIK3CA mutation. and we look to see what happens, and a whole lot of different combinations, and nothing works until we combine in brown the ketogenic diet and the PI3 kinase inhibitor, and this tumor completely disappears. However, in blue, the blue triangles are what if you use that same combination, but at the same time, every time you inject the PI3 kinase inhibitor, you also inject insulin at the amount that we typically would see in the bloodstream of the mouse had it not been on the ketogenic diet. So we're not causing ketones here or making them go away. We're just replacing the insulin that goes away when you go on a ketogenic diet. And now the tumor does not respond at all. So it's not the ketones that are killing the tumor, it's the absence of insulin that's allowing PI3 kinase to kill the tumor. So I think this pretty much nails it. I should say that it took us 1.5 years to get this published in Nature because the reviewers just were completely shocked by all the results, although in retrospect it's completely obvious that this is the way it should be once you think about what PI3 kinase is designed to do and what these mutations do. So I'll stop there. I just have some references which you can look at your leisure if you pull down the slides, that there's now a host of papers that have argued it's insulin resistance more than obesity that's driving cancer. And that's, you can see that if you have insulin resistance, ambient high levels of insulin, and a cell in your body picks up one of these PIK3CA mutations, it's now going to get all of the glucose in your body and it's going to grow. So I'm quite confident that that's why we see the correlation between insulin resistance. Obesity itself does not correlate. A nice paper by Strickler there on the middle one in this slide, showing that if you look at breast cancer, women who are overweight or obese but carry the fat peripherally and not in the liver do not have a higher rate of breast cancer. And women who are thin but insulin resistant have the same risk as women who are obese and insulin resistant. So it's really insulin resistance, I think, is our problem. The summary, I think I've already said all this. I think it's pretty obvious. I just want to get to the acknowledgments. So the paper that shows all this data, if you want to go into it, is just recently published in Nature in the summer. This was, a lot of this work was based on a clinical trial that we did with my Stand Up to Cancer team that's listed there as a second reference. And I just wanted, the third reference I put in for those of you who wonder how insulin stimulates glucose uptake into various tissues other than just muscle and fat where you have GLUT4. It shows that a protein called TXNIP, which degrades the glucose transporter, is destroyed, is degraded when you activate PI3 kinase pathway, AKT. And that probably explains the FDG-PET signal. So that's why that paper, I think, is really important. And I'll stop there. That's the questions. Robert.
Go ahead.
More questions than a full symposium. That was wonderful, truly tour de force, and in retrospect, yeah, why didn't we think of that? 3 questions, but I think they're all ultimately the same question. Get rid of the pasta.
Yeah.
Put back the apple juice.
Yeah.
Okay?
Yeah, apple juice is worse than pasta, I agree.
Yes, way worse, and the question is why?
Yeah.
So we talked just briefly yesterday about the fact that there are a whole bunch of sugar-dependent cancers, and they all seem to be endodermal in origin. In fact, female cancers, breast cancer, endometrial cancer, ovarian cancer, et cetera, liver cancer, pancreatic cancer, all from endodermal origin. They're not going through PI3, the fructose is not going through PI3 kinase, because that's a glucose transporter, not a fructose transporter. My question is, do you know if these tumors also have the GLUT5 transporter, number 1, which I do not know the answer to. Number 2, many tumors of endodermal origin have an enzyme called transketolase, which can convert fructose to glucose so that they could potentially power the cell irrespective of insulin. And is that one of the ways this works? And have you measured or studied transketolase in these? And the third one was this thing you just threw out at the end as this little throwaway, this TX-SNP. Because as you know, Richard Lee at Harvard has shown that TX-SNP is the thing that transports fructose across the intestinal epithelial cell, and it's self-induced. In other words, the more fructose, the more TX-SNP, the more TX-SNP, the more transport. And if you're blocking TX-SNP, could you be blocking sugar-dependent cancer, and is that a potential target? How many questions is that?
Yeah, we may have to answer some of these over a drink, but I'll say in short,
a very long drink.
Yeah, in short, GLUT5 is, we have a paper that's close to being accepted at Science that shows that in colorectal tumors, you definitely maintain GLUT5, so they can take up fructose from the gut, and that's actually helping them drive, and it probably explains why the 40-ounce Coke is driving your colorectal cancer growth, and probably explaining why we're getting colorectal cancers in the 30s, even the 20s occasionally now, which was unheard of because of the higher sugary drinks. The second question, so GLUT5 we don't see, we've looked at a lot of other tumors because like you, we thought maybe fructose is really driving a lot of tumors, but it's not. In part we know because very little fructose is in the circulating blood. It all gets sopped up. Whatever makes it into the blood pretty much gets sopped up in the liver.
Well, that's only true if you're drinking an 8-ounce Coke. If you're drinking a 20-ounce Coke, you way past hepatic extraction. And in fact, there's a fructose transporter on beta cells, which increase insulin secretion. So you can overwhelm your hepatic
I'm sure you're
extraction.
right about that, but yeah. So, but still, when we've looked at a lot of tumors, we're not seeing GLUT5 that often. TX-SNP, by the way, I think will also affect GLUT5 too. Every glucose transporter we look at, it regulates. So I think it also gets turned off by AMP kinase so that you can bring in glucose when you're energy stressed. Okay, so I don't know what other question I forgot to answer.
Well, so there was the GLUT5, there's the transketolase question.
Yeah, transketolase, we haven't really looked into that much. In these colorectal tumors that are using fructose to drive their growth, it's working by sopping up all the ATP and phosphorylating the one position. So they have ketohexokinase.
Yeah.
They phosphorylate the one position, but they don't have an aldolase that will hydrolyze, so it doesn't go into anabolic glucose metabolism, as we heard in the previous talk. So you can't make fats out of that fructose. It just stays there as fructose 1-phosphate, but it sops up enough ATP to allow the ATP shutoff of glycolysis to be eliminated, and now the glucose that's coming in with it. So the perfect storm is glucose plus fructose. Now the glucose is doing all the growth, but the fructose is acting catalytically to speed it up.
But why couldn't the fructose be used as carbon fragments for growth?
We thought it could be, but we did carbon tracing, and all the fat is being made from the glucose, even because we labeled it both ways.
It's not.
This is just colorectal. I'm not, I'm not saying others couldn't do it differently, but the colon, that's the way it works.
I see. Thank you.
A lot's being written about fasting plus chemo for cancer, or fast mimicking diet plus chemo. You're talking about a ketogenic diet. I wonder if you have any ideas on contrasting the two dietary therapies.
Yeah. So very nice talk given by Joshua Vinowitz. Joshua was also the one who had the paper on exceeding the capacity of the epithelial tissue, of the gut epithelium that we were discussing earlier in the previous talk. But Josh had a nice presentation at a meeting in Brooklyn last week, AACR meeting, 2 weeks ago, that tried this exact same thing that we did, except in pancreatic cancer, he was using conventional chemotherapy, not a PI3 kinase inhibitor, but he got exactly the same result. So keeping insulin down in the pancreas, and of course, those of you who are endocrinologists know this better than I, the ambient insulin level in the pancreas is always 10 or 15, maybe even 20-fold higher than in peripheral blood, because that's where it's made. And then downstream of that is the liver, which also has higher insulin than the rest of the body. So even though you never see PIK3CA mutations in the pancreas, they're still very dependent on PI3 kinase, and that ambient high insulin is keeping the pancreas going. Even with the KRAS mutation, you still need PI3 kinase. So I answered 2 questions, Robert's second question and yours, I think. And that makes sense? So I think that a lot of chemotherapy, there are papers scattered through the literature on either starvation diets or low carbohydrate diets effects on conventional chemotherapy in mouse models. But I've not seen a clinical trial published to prove that what you see in the mouse really is going to work in humans. But I may have missed it, but my bet is it will. And the problem is, I talk to the oncologists, no matter where I am working on my team, and I say, well, what are the patients doing during chemotherapy? They're sitting there in the IV. Well, they're taking Ensure because we don't want them to lose weight. Think about it. A very sugary drink while you're taking your chemotherapy. I mean, it's just crazy. So, who's next?
So my impression is this is mostly a discussion on treatment of cancers in patients who've already acquired mutations in PTEN or PI3 kinase. Do you have a sense of what's driving mutations in those pathways to begin with? And so do you think insulin resistance is contributing to that, and that people might prevent themselves from ever developing cancer by being on a ketogenic diet or a low carb diet?
Well, obviously for a mutation to occur, you need some kind of a mutagen. Probably most of the mutations that we're seeing in these tissues are due to reactive oxygen species. So
From hypermetabolism of glucose?
Hypermetabolism, mitochondrial electron transport problems are probably what's generating the mutagen. We're trying to figure that out because you can get signatures if you do full genome sequencing, you can get a signature of what the mutations are that correlate with either ROS or other antigens. You can get the nicotine effect, the ROS effect, et cetera. DNA mismatch repair defects, they'll all show up in the signature that tells you what's actually probably driven that tumor. But I don't think that keeping insulin down probably will help keep the ROS at a lower level, so you'll get less mutations in general. But a lot of mutations are just luck. Every time a cell divides, there's a chance of some mistake. And so even if we lived a perfect life, eventually some mutations would build up. I'm just saying that if you have high insulin, once you get one of those mutations, that cell's going to grow better than any other cell in the body. So you've got little microtumors that are getting driven by that high insulin.
Okay, thank you.
Some notables in the low-carb community have been developing a hypothesis of cancer as primarily a mitochondrial disease. And they refer to one famous study in JCI many years ago in which mitochondria or nuclei cytoplasm or nuclei were transplanted back and forth, cancer, non-cancer, and the cancer phenotype was reproduced based on mitochondrial transfer from a tumor, but not from the DNA. So one model relating to the last question could be that there's this reactive oxygen species that creates genomic instability. Is that consistent with what you know, and why would that instability, if the problems were originating in the mitochondria, why would they so consistently be hitting these 2 genes rather than, like all the other genes? I mean, reactive oxygen species shouldn't be selective, and we should be able to distinguish this hypothesis from, the classic DNA mutation hypothesis.
Yeah. So first of all, Warburg, when he saw this glucose uptake, he said they must be mitochondrial defective because they have plenty of oxygen. Why aren't they using it the way the normal tissue is? So that idea dominated cancer in the 30s, 40s, 50s, 60s, even when I was a graduate student. Mutated oncogenes didn't exist when I was a graduate student. Harold Varmus and Bishop's SARC mutation thing didn't appear until 1976, 77. So it was, everybody thought it was like what you said. It's just mitochondrial defects. It's a metabolic disease. We now know that there are, and subsets of kidney cancers really are due to a mutation in mitochondrial metabolism. IDH2 mutations, actually my lab in combination with Agios is the ones who figured how those work, the IDH1, IDH2 mutations. And so that, it does happen, but every cancer has a different way of evolving. It's just that when the PIK3CA mutation occurs, it allows that cell to respond to insulin better. And many other mutations don't. Most others don't. AKT mutations will also allow respond to insulin, and PTEN loss will allow them to respond to insulin. So it's frequent, but it's not invariable. Many other paths that can be taken.
Given the availability of exogenous ketones, could you imagine a chemotherapy in which you develop an antibody to insulin and basically shut down insulin, in that way, and then just support the patient with ketones?
It's an interesting idea. I think that I love the idea that ketones can replace the majority of, or at least half the glucose requirement in the body. And I particularly like this because I always like to think about everything in terms of evolution. And if literally metabolically, we're not a whole lot different today than we were 50, 80,000 years ago. A few differences. We can maintain the ability to metabolize lactose with age. That's the biggest mutation we see. But most other things we haven't changed a whole lot in metabolism. We were hunters and gatherers, mainly hunters probably, and what we gathered. Even before we had fire, we didn't cook anything. So if you think about all the things we learned today that were healthy, they were the things that were eaten by cavemen 80,000 years ago. Uncooked vegetables, roots. The only time you got fruit was like 1 month out of the year if you lived in a temperate climate. So any human beings that lived in a temperate climate had to figure out how to survive for long periods of time without any food. So in some ways, we took the same evolutionary path as the bear. So bears go to an extreme. They not only slow down their metabolism, they actually go to sleep. But before they go to sleep, what do? They wander through the woods and they eat every piece of fruit they can find. Somebody once told me they eat something like, the average bear eats some 30,000 blueberries in Maine before it hibernates. They will climb trees and eat honey in spite of being stung all over the place. They'll still eat that honey. And they'll do it in the fall, right at the end of the season. And they put on 100 pounds or more in a month. And then they become severely insulin resistant, slow down their metabolism, fall asleep, and there's enough fat stored to make it till the next season where there's still roots now available they can dig up and have something to eat again. We're kind of like that. That's why we become insulin resistant. And that's why we love sugar. We love sugar because 80,000 years ago, any human being that didn't go through the same cycle as a bear and put on 20 or 30 pounds at the end of the growing season by eating every fruit in sight and becoming insulin resistant and slowing down their metabolism was going to die of starvation before the next root was available when the ground thawed out 3 months later or 4 months later. So that's why we love sugar. It's, people died of starvation. They didn't die of obesity. It's just that we now have all that sugar available all year round. So I know there wasn't a question about that. I just wanted to say it.